We're at the CROI in Seattle. We're here with Deanna Kulpa, who is with Emory University, and she is going to be speaking on basic science, which is something everyone really needs to know a little bit about, and because that's a large part of the conference.
Mm-hmm.
You kind of are working your way up from the bottom of the iceberg. You know, the bottom is the iceberg is with all the little things that we need to know to get to the to break the surface
mm-hmm.
and come to a point where you actually have something you can work with in clinical trials.
Mm-hmm.
So here we are at somewhere around under the sea, working on the basic science, and you're working on CD8 suppression.
Mm-hmm.
So tell us a little bit about that for our audience, who is many of them not scientists, but some of them are. So
mm-hmm.
we'll take it from there.
So so we know that in HIV infection, right? So HIV infects CD4 T cells,
mm-hmm.
and we know that this is a persistent infection, that we have drugs that do very well at reducing viral replication in CD4 T cells down to undetectable levels, right?
Mm-hmm.
So then the question now, when we're thinking about basic science, is really thinking about well, what do we do with the cells that are left after we take the drugs and we reduce the viral loads down to undetectable levels? There's still CD4 T cells that have virus in them. It's just not
mm-hmm.
showing up,
mm-hmm.
right? And so we want to know, well, how do we get rid of those cells? So there's been a lot of really exciting work in thinking about how to induce or excite the CD4 T cells to start expressing the HIV.
Out of a compartment,
yeah,
so speaking.
Out of latency, to be able to recognize those cells and find them and kill them. So the frustrating thing has been is that when these drugs have gone into clinical trials to try to get the CD4 T cells to start to express HIV so we can target them and kill them, they haven't worked very well. They haven't
mm-hmm.
done their job of
mm-hmm.
getting the CD4
getting
T
all of it out.
of getting all it out.
Yeah. Mm-hmm.
So what we've been studying and realizing is that the body, the immune system, has adapted ways of kind of controlling excitement of the CD4 T cells in
mm-hmm.
a sense,
mm-hmm.
because a lot of times inflammation is bad for us,
mm-hmm.
and so we actually
so there's a redundancy built into the body's normal mechanisms.
Exactly,
yeah,
and
yeah.
so we have CD8 T cells that kind of come in and say, no, you guys got to calm down. We need to kind of keep things under control. So the drugs are trying to get the CD4 T cells to wake up and express the virus, and we're actually finding out that CD8 T cells have some mechanisms that come in and actually putting the brakes on all of that.
Mm-hmm.
And so this is part of the reason we believe that some of these drugs aren't working as well as we think they're going to once you start putting them in people, is because there's lots of mechanisms out there that are really about controlling the inflammatory process in
mm-hmm.
people. And our research is really trying to understand, well, exactly what are the CD8 T cells doing? Like, what are the pathways involved? Because if we could understand that, maybe we could actually intervene and say, we like the good parts of the CD8 T cells that can kill cells, the cytotoxic T lymphocyte activity that can target HIV-infected cells, get rid of them, but we want to get rid of maybe the parts that are putting a brake on what we're trying to get at, which is waking up the CD4 T cells. So keep the good, get rid of the parts we don't want, and can we go through an intervention that way? So our basic research is trying to understand the mechanisms, and our group partners with investigators at the Yerkes Primate Center who are doing in vivo studies using the monkey model system.
Mm-hmm.
So we work very closely with them to help to say, well, what sort of interventions can they try in the animal models,
mm-hmm.
because we're very translational in thinking, like, how do we then push this into the next level? We can actually try to get an intervention that would work in people.
And just to know that the CD4s are like the innate immune system.
Mm-hmm.
That was from way back. That's basic.
Mm-hmm.
The CD8 is adaptive immune
Right.
system, which it acquired a knowledge of how to work,
Mm-hmm.
and it learns different things over time with different diseases and so forth. But the HIV virus is so confounding, and then you also know that we have to have a little bit of inflammation.
Mm-hmm.
That's part of what we have to have. So working in your field it's complicated.
So what we're finding is that the CD8 T cells also have an innate mechanism. So we think about the CD8 having the adaptive, what you're talking about is actual recognition, the specific recognition of, this cell is infected with this particular virus. Oh, I need to target that, right? But we're actually finding that that
Innate
CD8
built
T
in.
also have a innate mechanism where they can non-specifically just come in and say, we can secrete some cytokines, make some contacts that help to regulate, because there are lots of regulator CD8 T cells that it could kind of come in and say, we like inflammation, just not that much inflammation, or just try to kind of tone it down, or control inflammation response.
Right. So your outcomes are how? How do we use your outcomes?
So so right now, when trying to identify the mechanism, really what we're trying to do is try to find signaling pathways. And what's really great about Emory is, so there's so many wonderful investigators. So I work with Dr. Raymond Shanazi, who's a chemist who's has a huge team that we can work with. That if we can identify some signaling pathways, it would be wonderful to be able to then target those pathways in some sort of chemical modulator. There's something that we could actually say, okay, what do we do about this particular pathway that's suppressing the CD4 T cells? If we need them to wake up and start expressing, like I said, we want to keep the good, the adaptive, immune response of the CD8s, but maybe change how the innate response is working, maybe against some of the LRA or the latency reversing agents that we're trying to apply in the clinic.
It's exciting, and I think the other thing we like to do is, if you find as an audience member this exciting, especially if you're a young person, you might want to get into the field, because it sounds to me really exciting. I know that a lot of people say, well, science is boring, but when you come upon stuff like you're discovering now, things that I did not know just till you mentioned them just now, it's important. These are all small building blocks to take you on this ever-growing steps, multiple steps that get you to answers that can actually be included in trials, and or a sense of how we build a theory that then turns into an outcome that is very useful. So, and that's very important in the cure, is trying to find out how we can work with some of the building blocks to end up with a cure.
I think one of the things that has always fascinated me about immunology and HIV in particular is, one of the things we've learned as we've tried to tackle the virus and how, what we need to do about it, is it's forced us to learn more immunology and more about how the immune system works that we didn't know before, until we started to try to figure out what is happening with this virus and how it's getting around the immune system. And the more we learn, the more we realize, wow, there's still more we got to go. And I think what's cool about being a scientist is you get to be the person who figures out what we didn't know before. And that's part of what we do, is learning things that literally no one else in the world knew until we discovered them, and getting to be a part of that, and then also thinking this has the potential of helping people and really providing a foundation for how we can actually make some real meaningful change. It's also part, what is exciting about it as well.
And so the hall talk where you just gave a presentation says, wait a minute, I needed that piece of information. So now you're doing a collaboration because you're working further into that area, and they need what you have. Collaborations blossom. And that's what we call the hall talk,
Yes.
which you couldn't have if you didn't have a conference, because you have to bring everybody together to we call it peer review, which is you're looking at everybody else and you're kind of trying to shoot holes in their conclusions. Well, not nasty like, but just saying, how did you really get to this? Quiz them on it. And then at the same time, they're saying, wait a minute, I really need this piece of information, so we need to work together. I need more information. Because everything you present is not everything you know.
Right, oh, absolutely.
You really do have a wealth of knowledge that you've learned that probably wasn't in and of itself worthy of the presentation. And you just have so limited time and space on a poster. But it's important to know that you are working in that area and you've got a lot of answers. So we really appreciate your giving some of that, a sense of it, to our community about basic science. That's really important, and we thank you for that.
Oh, thank you. This is a lot of fun.
Thank you.
Thank you.